Hello. So we are going to talk today of human pathogens, and I will try and split my talk into three parts of 30 minutes each. And we will try and discuss issues of drug resistance and immune evasion, and also talk a little bit about the epicoplast, which is an interesting organelle of the malaria parasite, and see how that can be used as a new target. So I will share my slides now. I hope you can see the presentation. So basically, I'm going to talk about immune evasion and the drug resistance issues in human pathogens. And the pathogens we will basically talk about are parasitic pathogens, a little bit also on bacteria in the beginning. So we want to understand what are the new drug targets that have been looked at and the challenges of drug resistance. Also immune evasion for survival and growth. So what are the evasion strategies that parasites adopt to evade the human individual immune response, and also what issues and challenges this poses for development of vaccines. To also look at the epicoplast, which is an allelic plastid in the malaria parasite, and how this can be an Achilles heel for malaria because it can offer avenues for intervention against the malaria pathogen. So if you look really at the way antibiotics have been used, and this is of course for bacterial infections, one can see that so if you look at the introduction of penicillin in the 1940s and how penicillin resistance was first, started being seen in pneumococcus, and the first report started coming in 1965. But then tetracycline resistance was, within 9 years, you were reports of tetracycline resistance and Shigella coming up. So here you have the antibiotics introduced and the years in which they were introduced. And on the left side, you have this, think of where the first resistances to these antibiotics started being seen. So really a bit introduction of an antibiotic, and within several years of the introduction, you start seeing resistance developing in the field. So there is blood pressure, because of which there is selection of resistant pathogens, and then these pathogens take over and become an issue, and you have resistance developing. So a single, so a bacterium can be resistant to a single drug or to multiple drugs, and or can have drug resistances which are called, MDRs, multiple drug resistance, or XDRs, to resistance to a large panel of extensively drug resistant drugs, bacteria. Or you can also have what is also now called TTR, although the instances of TTR are not so much. So basically, this becomes a very, very important problem, and not just that. And you must have heard of these bacteria called escape pathogens, which is a panel of bacteria that you see in hospital infections, and there are big issues in handling bacterial infections because they have so much resistance to known antibiotics. So this issue, I'm just putting the slide here because I wanted you to see how important the issue is. And also this issue of resistance is across the field. It's not only seen in microbial infections or in fungal infection, but it's also seen in parasitic infections. And when I say parasitic infections, I take some examples. And the examples I talked to you today about are basically malaria, which you see here. You have malaria parasites infecting an RBC. This is an leishmaniasis, leishmania infection, where the leishmania parasite, which is a kinetoplast, infects the cells of the humans. And here you can see a worm, which is a filaria worm, which causes an elephantiasis. And this is also a parasitic infection. So basically, we can take examples. Either you take examples of malaria or leishmaniasis, or other parasites like toxoplasma, which is an opportunistic pathogen. When you have immunocompromised individuals such as HIV patients, you can have toxoplasma infections, you can have cryptosporidium infections. These are very important and cause diarrhea in children. And in the eastern part of the country, this is a very important parasite. So basically, all these parasites have drugs that are given as cure for these diseases. But there is resistance developing against these drugs. So if you look at chloroquine, for instance, which is an important drug given for malaria infections, chloroquine resistance in Plasmodium falciparum became a big problem. And therefore, there was a complete shift in the treatment to what we call artemisinin combination therapy. Similarly, for leishmaniasis, early on antimonials were given as for leishmania treatment, but then because of issues of resistance, etc., and also some issues of safety, leishmania drugs profile treatment changed to miltefosine, and now it's amphotericin B in liposomes. So basically, and resistance keeps coming up even in these parasites, perhaps not as a rate as fast as bacteria develop, but still it's pretty fast. And so the world has to keep pace with developing newer and newer compounds for countering resistance in parasites. So as an example, most of my talk, I will delve with malaria, and therefore, this, the infection cycle of malaria, but it's very important to go through it. Remember this, because when we talk later on of the issues in malaria infections, this is what we will be, alluding to time and again. So if you look at P. falciparum and P. vivax, which are the major parasites causing infection in humans, apart from malaria, ovaria, and olecine. Falciparum causes severe infection, can also cause death. Vivax is important because it causes significant morbidity, but also because there is relapse associated with disease. So you get a recurrence of infection. So this is a problem with Plasmodium vivax, because Plasmodium vivax can stay as hypnozoids in the liver, and these can get activated. That's that's probably the way that happens. In any case, if you look at the life cycle of the parasite, it is a generic life cycle. You can see, that the mosquito bites and the mosquito carries sporozoites, right, in the salivary gland. And these sporozoites then traverse the skin and they reach home into the liver. And then once they home in the liver and infect hepatocytes, they multiply extremely fast in hepatocytes. And each infected hepatocyte can actually give rise to 10,000 to 30,000 parasites plus per infected hepatocyte. So this is really, really, the number of parasites in the liver is very high. Now, interestingly, you do not see any symptoms. There is no obvious fever or anything when the person has an infection in the liver. It's only when the hepatocytes rupture and the merozoites are released into the bloodstream and they start infecting the RBCs that the symptomatic phase of the disease starts beginning. And you hear when they infect the RBCs, they develop here into a small wing stage, then they become, trophozoites, and then the trophozoid stage, it multiplies, gives rise to many, many merozoites, and then this RBC ruptures. And these merozoites are released. And it is at this time that the chills and the fever, etc., start happening, and the toxins of the parasites are released into the bloodstream. And these merozoites will again cause a fresh cycle and infection, and this keeps on. A part of the parasite gets converted into gametocytes by the gametocytogenesis. They convert from the gametocytes to the macrogamete, which is the female gamete, and the microgamete, which is the male gamete. There is fusion into the zygote and the formation of ookinete, which then goes into the midgut wall of the mosquito, develops into an oocyst, and here division into further daughter cells happens. And these then form sporozoites, and sporozoites are then again transported to the to the salivary gland, and the salivary gland sporozoites then mature, and then these are transferred back into the humans. So the cycle then is between the Anopheles mosquitoes and the human. So we must remember, therefore, that the time taken from, the pre-patent period, which is the time taken from the liver to the blood stages, is called the pre-patent period of this, and the symptoms are basically here in the erythrocytic stages of infection. This is the symptomatic stage for disease. Now, so malaria, as we know, is characterized by fever, headache, and chills, and there is also sweating. And you can have recurrent fever, and the duration of the recurrence of the fever is dependent upon the species with which a person has been infected. It is due to the release of parasite toxins into the bloodstream, which happens during the, as I mentioned earlier, erythrocytic cycle or the RBC cycle of merozoid egress and reinvasion of fresh RBCs. As infection progresses, the number of RBCs keeps decreasing because you have infection and the RBCs lyse and the merozoites are released. So there is actually, if the infection levels are very high, if the parasitemia in the person parasitemia is very, very high, then the lysis of the RBCs is so high that the rate of formation of new RBCs cannot keep up with the lysis of the RBCs, and this can lead to severe anemia. RBC containing parasites such as RBCs that contain parasites such as Plasmodium falciparum can also sequester in deep tissues, and this is a very interesting phenomenon because so if you have a parasitic infection, so in the RBCs, the parasite takes in a lot of proteins and other material from the RBC. But it also secretes a lot of proteins into the RBC, and some of these proteins then go into this membrane of the red blood cell. And when they do that, they also cause certain modifications in the RBC membrane. And one of the modifications, a very obvious modification, is formation of certain protuberances called knobs. And this is primarily because of accumulation of the protein called PFEMP1 and some other proteins, but primarily PFEMP1, which helps in the formation of knob-like structures. And these are very important for cytoadhesion and interaction with the endothelial cell proteins. And I'll come to that cytoadhesion issue a little later. When that happens, this can actually cytoadhere to endothelial cells. And because of this, there can be, this is one of the causes for cerebral malaria, because you can have cytoadhesion in the microvasculature. And this can actually affect blood flow into the brain, and you can have cerebral malaria. But this can also have consequences in other organs, and this can lead to multiple organ failure. And if that happens, then the pathology is very severe and can actually lead to death of the individual. So this is how you can have an extremely severe disease. Now, to understand how treatment can happen for infections, it's important to understand the parasite and what sort of structure and what sort of mechanisms and the basic major targets. And I just give you 2 or 3 examples from this figure from a paper from 2019. So this is an RBC, outer one, and this is the infected, this is the parasite inside an infected RBC, a single parasite in this case. And you have a nucleus. So this is a parasitophorus vacuole, inside which is the parasite. The parasite has a nucleus. The parasite also has organelles such as a mitochondrion. It also has, so it has a single mitochondria, and it also has a single epicoplast, right? And this epicoplast is a lilic plastid-like structure. I will describe it later. Now, if you are looking, this is the digestive vacuole. Now, in the case of drugs for malaria, this digestive vacuole is very important. So what happens is that hemoglobin is taken in by the parasite from the RBC, and a lot of hemoglobin comes into the parasite. It's internalized. And the hemoglobin gets broken down into the globin and the heme components. But the heme component on its own is actually extremely toxic for the parasite. So it must be converted from heme to hemozoin. And if that conversion is not happening, then the toxic heme can cause parasite death. So what chloroquine does is, chloroquine actually interferes, a drug, with this conversion. It actually stops the conversion of heme to hemozoin. And by doing so, because the parasite is not able to convert heme into a harmless hemozoin pigment, the parasite dies. Similarly, piperaquine does the same. The other drug, atovaquone, actually inhibits cytochrome B, which is a part of the electron transport chain of the mitochondrion. And this is a direct target for the drug. If there's a mutation in cytochrome B, in the binding site or other sites that affect the binding site, then atovaquone will not be able to bind. And if that happens, then you have resistance to atovaquone developing. Similarly, you have sulfadoxine pyrimethamine. Sulfadoxine pyrimethamine target DHFR and DHPS. If they have mutations in DHFR or DHPS, which affect sulfadoxine or pyrimethamine binding, the resistance to these drugs develops. Now, this is the way the target of the drug can be mutated or modified to affect the binding site of the drug and therefore give resistance to the drug. However, in the case of chloroquine and piperaquine, primarily in the case of chloroquine, piperaquine I'll come to later, what happens is that the target is not mutated. What happens is that the transporters such as PFCRT here and PFMDR1 on the surface of the digestive vacuole can pump out chloroquine, right? So the efflux of chloroquine out of the digestive vacuole can decrease concentrations of chloroquine here. And certain mutations of PFCRT and PFMDR1 actually cause a large efflux of chloroquine. And when that happens, then the chloroquine concentrations cannot build up in the digestive vacuole, and because of which chloroquine is unable to act. So it's not a result of the mutation of the target site, but rather a mutation in the efflux pump, which causes excessive efflux of the drug out of the digestive vacuole, lowering its concentrations and therefore affecting its activity and causing resistance to the drug. Now, this is a very crowded slide, and I'm sorry for this. But basically, it tries to tell you what kinds of resistances are encountered. And this is a continuation of the previous presentation, previous slide that I was talking about. Now, after chloroquine resistance came in, there was this new class of drugs that was introduced called Artemisinins, also from Artemisia annua. And you know that a Nobel Prize went into the discovery of artemisinin. They caused a lot of improvement in drug therapy for malaria, and many, many millions of lives are saved. The current primary therapy uses artemisinin combination. So artemisinin derivatives are given in combination with other drugs. So it's called ACT, artemisinin combination therapy. Right? So you can have artesunate or artemether, other artemisinins, and RDHA, dihydroartemisinin. These are first-line treatments for primordial falciparum. Now, mutations in KES13 and K13 started coming up in Cambodia, Thailand border, and one started seeing delays in the response to artemisinin treatment, which essentially meant that parasites were developing resistance to artemisinin too. Now, this is a very serious problem, because if these resistances spread, and they will spread and are spreading across the world, then the resistance to artemisinin drugs will come in, and these combinations will start failing. So what is the follow-up? Do we have new compounds and new drugs to take over ACTs in case the resistance is spread and ACTs start failing? Similarly, if you look at chloroquine, and I mentioned this earlier, the chloroquine heme detoxification happens in the digestive vacuole. That's the affected pathway. And mutations in CRT and NDR1 actually cause resistance. And mutant PFCRT in Africa is less fit. And it has been, so what happens is that when you withdrew chloroquine from treatments, it's in Africa, for instance, then the wild type parasites took over. So what happens, so this is an interesting observation that so chloroquine was being given, chloroquine resistance developed, then chloroquine was withdrawn from treatment programs, right? And many years after this, it was evaluated whether the parasites now seen in this part of the world actually had resistance. And it turned out that no, once you withdraw the blood pressure, when you withdraw the treatment, then wild type parasites are taking over again. So there is still a possibility can then chloroquine like molecules be reintroduced into treatment profile, because now your resistances have gone back. The parasites could not survive. The resistant parasites were not as fit as wild type parasites. And once you withdrew the blood pressure, the resistant parasites were not as good as expanding and infecting, and therefore wild type parasites would take over. Take the example of piperacin. Piperacin is also an aminoquinoline like chloroquine, and it is a partner drug for in ACTs. It's given with dihydroartemisinin and piperacin as a combination. This was really one of the frontline treatments given in recent years. And this also targets heme detoxification. It has piperacin. The resistance to piperacin did come from PFCRT mutations, but it seemed that primarily resistance came from amplification of plasmapin 2 and 3 genes. And this resistance was not solely because of PFCRT mutation, but also because of plasmapin 2 and 3 gene amplifications. So this is another mechanism by which resistance to aminoquinoline could come up. Similarly, lumefantrine. Lumefantrine is an aryl amino alcohol. It's a partner drug for ACT again. This also might include inhibition of hemoglobin import or heme detoxification as its target. And PFCRT mutations, PFMDR1 amplifications, and sequence of mefloquine LMF have an effect on the resistance of this drug. And similarly, MDR1 overexpression imparts fitness cost. So all these resistances have fitness costs, and this also has implications on whether you can actually withdraw drugs and then reintroduce them to counter these. Now, what is alarming in a sense for malaria is that although the number of the cases of malaria cases in the world has come down, there have been eradication elimination programs, some very successful, such as the elimination program in Sri Lanka is really an example for India, because Sri Lanka was, even in deep political crisis and problems, was able to curtail malaria to a large extent and was declared malaria free some years ago. So it's possible to, and numbers have come down. And in India too, Orissa has shown a lot of improvement in bringing down malaria cases because of very targeted intervention strategies. But still in Southeast Asia, if you look at the contribution of India, and if you look at Southeast Asia, India contributes 88% of total malaria cases, and really 91% of PY-VAX infections, nearly 89 to 90% come from India. So it's also quite a center for PY-VAX infections. What is alarming for treatments, and as I mentioned this earlier, is that the latest frontline treatment, which was in the field, DHA piperacin, resistance to DHA piperacin started being reported in 2019. And the first paper showed that when they gave DHA and piperacin combination, there was 50% treatment failure in Southeast Asia. And this is a very, very high treatment failure, which essentially means that you have resistance to DHA, to piperacin developing, also affecting the resistance to an artemisinin drug. And this can take down the first-line anti-malarial in this part of the world. So this poses a great threat to us. And because of this threat, it's very important now to understand that newer and newer drugs have to be constantly looked for, and constantly we have to start looking at new targets. Now, when one says targets for a particular drug, what must a feature of a target be, right? A target is a protein, primarily a protein that's important for parasite function, not just important, in fact, it should be critical for parasite survival. And therefore, if you are targeting this protein, or your drug has multiple targets, or the multiple proteins that are being targeted by the drug should have very important effects and should have very critical effects on parasite growth and survival. Moreover, if a drug is targeting certain proteins in the parasite, the drug should not target similar proteins or similar domains in the human host, because if it does that, then it will have a lot of safety issues and toxicity of the compound will be high. Therefore, it's not a trivial problem. I mean, developing compounds, it's not enough to identify a good target. Of course, the target has to be critical. The target should be essential for parasite survival, but one should also make sure that these proteins have domains, all these proteins are absent in the human host, so that, you can have drugs being identified for these. Easier said than done. Target-based drug discovery has been done, for molecules now for malaria, but this is a phenomenon only that's come up in the last, maybe decade or more. Earlier on, both chloroquine, artemisinin, etc., were developed as compounds that came out of screening strategies, which were not target-based. The targets of chloroquine was discovered much later, much after the drug was discovered. So anyway, a lot of work has gone into identification of targets, and some leading targets that we have today, and this is not an extensive and not an exhaustive list, but just to give you some examples of targets that are being used today is here. So as I mentioned earlier, when we were talking of sulfadoxine pyrimethamine, DHFR, dihydrofolate reductase, is an important target. Similarly, you have in P. falciparum ATP4, which is an ion channel. So ATP4 inhibitors have shown very good efficacy, at least in vivo, and these are now being taken forward in clinical trials. So the evaluation of these molecules is first, any of these molecules. If you have drugs or small molecules being made against any of these proteins, and they're first evaluated in vitro culture systems, then they move into animal models or humanized mice that are used as models to test, in which case what one does is you can either have a Plasmodium yolli or a Burghain mouse model system, or what is gaining prominence now is a system in which you have a nude mouse, an immunocompromised mouse, in which you actually put in human blood, and then you can have a P. falciparum infection, because you can't see a P. falciparum infection in mice. It doesn't infect mice. So you can actually take immunocompromised mice, put in human blood, circulate human blood, and give a falciparum infection. So basically, it's called humanized mouse model. You can evaluate drugs and do their PK/PD, pharmacokinetic analysis, their pharmacodynamics in a mouse model. Anyway, so many systems exist, and these kinds of evaluations can happen. And based on these, one identifies inhibitors. But the targets have to be so the targets have to be worked out in great detail to see that the targets against which you are designing compounds are actually worthwhile, and the resistance to these will not develop early, not develop fast. So basically, if you have a target that you design a drug against, and if the target can mutate very fast and the active site, the binding site of the drug can get altered extremely fast, then there is a great likelihood of resistance generating very fast in the field. So all these experiments also have to be done to understand how fast mutations can develop against particular molecules. So all these become part of the discovery process. Anyway, coming back, so some leading drug targets then for malaria are DHFR, dihydrofolate reductase. You also have PFATP4, which is an ion channel. Then you have a long-acting, you can have an ATPase as a target, and there is a long-acting ATPase inhibitor that's currently under development. There's also an elongation factor 2, it's a translation factor 2 inhibitor. Then there are inhibitors for DHODH. This is a mitochondrial enzyme, dihydroodorate dehydratase, and there are inhibitors for that which are under trial. And they also have epicoplast-specific inhibitors. Now, in the epicoplast, you have a pathway which is called the isoprenoid biosynthesis pathway, and this follows a non-mevalonate mode of isoprenoid biosynthesis, and this is really essential for parasite survival. And if you mutate or knock down any of the enzymes of the isoprenoid biosynthesis pathway, so ISPG, ISPH are all enzymes of the pathway, then the parasite dies. And so basically the epicoplast then becomes essential for parasite survival. And the main reason for this is that the isoprenoid synthesis is affected and the parasite is not able to survive. So ISP enzymes are very important targets for development of drugs. And there is an inhibitor of ISPs called phosphamidomycin, which is currently under trial with clindamycin. So epicoplast is an important target, particularly the ISP pathway of the epicoplast. Similarly, you have other approaches, like you can have heparin-derived polysaccharides that prevent cytoadherence. So this can actually decrease the morbidity of disease and cause the effects of P. falciparum that you see in infection can be addressed here. So there are others as well. I'm just giving you a glimpse of the kinds of targets that are in use, and one hopes that many of these, or at least a few of these, will show success, and that we will have new inhibitors that can help us address the problem of artemisinin resistance that is coming up and is so dangerous now. So if one looks at, and I'll end with this slide to not give you an overdose of drugs, but basically this tells you the kinds of efforts that go across in the world. Now, when you talk of drugs really for infectious diseases, for diseases like malaria, leishmaniasis, filariasis, big pharmaceutical companies are very rarely interested because these are diseases of the developing world, what was called the third world, and primarily. And the purchasing power, these drugs can't be expensive. These are basically part of public health programs. So therefore, these are not profit-making enterprises and cannot become something that big pharma will be interested in. Therefore, many groups, several non-profit organizations, national laboratories, public health, individuals, and work on developing of new molecules. And one such organization is the Medicines for Malaria Venture, and this is a not-for-profit organization that really coordinates efforts worldwide to bring out many molecules and to see how they can move fast in the development mode. Now, when one talks of drug development, it's very important to understand that drugs do not come, cannot be developed extremely fast because there are several steps in the development process, and these are essential to help us establish whether a drug molecule is not only efficacious but also safe, and to the correct dosing of the molecule to ensure safety. So you have an early phase, and I showed this slide for that particular purpose. So you have a preclinical phase where you have the evaluation of these molecules happening in the laboratories and on animal models, etc. And then it moves into the human volunteer phase, which is basically a phase 1 trial to try and understand whether this drug is safe or not. And once the safety of the molecule is established, start moving into a phase 2 trial where you start seeing the efficacy of the molecule in terms of the disease, whether it is being able to basically work function as a drug. And once you do that, you move into a large scale phase 3 trial. And then once all this is over, you basically do a regulatory review, and then companies have to start producing it and marketing it and so on and so forth. So as you can see here, there are several molecules that are under development in various phases of development. You have somewhere you have a translation phase, preclinical phase where human volunteers are now coming into recruitment and different drugs are being tried for malaria. Then again, you have patient exploratory phases where you have a phase 2 trial sort of coming up, and in that 2 different phases of the trial are happening. And then you have a phase 3 where certain combinations are being tested, right? And then of course you have access and approval, wherein these drugs are then approved for use. I did not mention one thing when I was talking of drugs for malaria, and as I mentioned the fact that P-Vivax causes relapse. So if you use these compounds against Vivax, they can help you cure the symptoms at that particular point of time when a person is showing symptoms of Vivax infection. But how do you prevent relapse from happening? Because if you prevent that, then you must have a molecule that is able to affect the liver stage, the hypnozoid stage that actually is responsible for activation at a later time for relapse. So basically there's a drug called Primaquine, which is an 8-aminoquinoline, which has a which is able to kill P-Vivax hypnozoids and have an effect, and therefore prevents relapse. And it's a 14-day treatment given correctly as a 14-day treatment course. But the problem with Primaquine is that in individuals who are G6PD deficient, it causes a problem of hemolysis. So if a person is G6PD deficient, and this happens, this can be a problem in certain tribal belts in India, then there is a whole risk of hemolysis, and it's going to be very dangerous for the person. So when you have to give Primaquine, you really have to test whether the person is G6PD deficient or not. And also the treatment is quite long. So now there is a new drug called Tafenoquine, in which the treatment has been reduced. It's a single dose, just one tablet has to be taken orally. So the treatment time has come down from 14 days to 1 day. But the problem of G6PD deficiency and hemolysis have not been resolved. So basically, when you give Tafenoquine, even with Tafenoquine, you have to test whether the person is G6PD deficient, and if the person is G6PD deficient, then you cannot give Tafenoquine. It's contraindicated. So this problem remains. So one of the challenges for Vivax drugs is also to have a medicine or drug that can not only kill the hypnozoids and therefore prevent relapse, but also have a molecule that does not cause hemolysis in G6PD deficient patients. So this is an important issue that also needs to be addressed. So I'm just trying to tell you the kinds of complications that exist, and that there are many issues that have to be resolved in the drug discovery process. This is as far as I wanted to say for the first part of the talk. And just to take for you to take home, the complexities of the drug pipeline for infections. And in the next 30 minutes after this is one recording. In the next recording, we'll try and address certain issues that the malaria parasite deploys to overcome the immune response. So what are the strategies for immune evasion that are adopted by the parasite? And we will do that in the next presentation.